The last thing we're going to talk about are some of the immune system disorders. And the first thing among those disorders that we're going to address is something called hypersensitivity. Hypersensitivity, in essence, is basically any kind of over-response the immune system has to an antigen. We can call that antigen specifically an allergen if it's going to induce some sort of a hypersensitivity response. The key feature here is that, okay, it's fine that my immune system responded. It should respond, okay? Immune system responds, that part's normal, but what is abnormal is that the immune system responds so vigorously that it results in tissue damage, which is something we do not want. So hypersensitivity reactions are immune system reactions that also promote damage to tissues. And there are a number of different examples of hypersensitivity. The one most of you are familiar with is allergies. Allergies can be very mild, like having a hay fever allergy or whatever, or they can be very severe. Some people, for example, are so allergic to bee venom that if they're stung by a bee, they can go into anaphylactic shock and die. Sometimes hypersensitivity reactions can occur, actually they frequently occur, I should say, not sometimes, but they can frequently occur during organ transplants. This is called alloimmunity. Allo means different, so your body is responding to a different cell type that's been introduced. And we know that it's a different cell type because your T cells notice that, hey, this donated organ has MHC1s on the surface of its cells that are shaped differently than what I would expect, so it must be foreign and I should attack it. Hypersensitivity reactions also include autoimmune diseases, things where the immune system erroneously or wrongly identifies parts of your own body as being a foreign agent. Sometimes, for example, some people's body identify their own DNA as being a foreign agent and begin to attack it, and that can cause a development of a type of autoimmune disease called lupus. And there are some other examples of autoimmune diseases that I will discuss later, just briefly. There are four main types of hypersensitivity, four ways in which we can induce tissue damage via this immune response. Type 1 hypersensitivity reactions are the ones that are mediated or regulated by immunoglobulin E, antibody E's. This is a type of reaction that's often called acute hypersensitivity, and it's the kind of reaction we see occur in people who get anaphylactic shock from, say, a bee sting, or if they have a severe peanut allergy or whatever it might be, and they go into anaphylaxis. The reason for this is that those immunoglobulin E's, through a process we are not going to learn about, but those immunoglobulin E's cause mast cells and basophils to degranulate. And when they degranulate, they release huge amounts of histamine throughout the body. You'll remember histamine does two major things. It causes vasodilation and it induces increased capillary permeability. The combination of these two events happening systemic-wide immediately is that your blood volume drops, the vasodilation causes your blood pressure to drop, and you can go into hypovolemic shock. Type 2 hypersensitivity reactions are a little different. Rather than involving the response of immunoglobulin E's and systemic-wide degranulation, in a type 2 hypersensitivity reaction, we have antibodies responding to antigens that are present on a cell. The antigens have got to be stuck onto a cell to be a type 2 reaction. And if we have antibodies that are shaped correctly to respond to those antigens on that cell, we have, by definition, a type 2 hypersensitivity reaction. A great example of this, classic example of this, is what the picture is showing here, blood transfusion reaction. You give somebody the wrong type blood, and the antigens on the surface of the donor blood are going to be attacked by antibodies in the recipient's blood. And you're going to get those agglutination responses you've seen. So antibody-dependent cytotoxic hypersensitivity is a type 2 reaction. The third type of hypersensitivity reaction is called the immune complex hypersensitivity reaction. In this case, rather than having the antigens being attached to a cell, as we saw in type 2, the antigens are free-floating. So, for example, a person who has lupus, like this poor woman here in this picture, they have pieces and parts of old dead and dying cells, like DNA from some of their dead and dying cells, and or other proteins found in those dead and dying cells, and their own body mistakenly identifies those DNAs as foreign antigens that need to be attacked, and this induces a very severe inflammation response, usually in places like connective tissue. If you're looking at a lupus patient, you can see this person's face is quite inflamed, and it can cause tissue damage, as what a hypersensitivity reaction is by definition. Usually with type 3 reactions, there are 3 types of organs in the body that are most severely impacted by this process, and those include the joints, the blood vessels, and the kidneys. Now, a type 4 hypersensitivity reaction is also called a delayed hypersensitivity reaction, and that is because oftentimes it takes a couple days from the time of exposure to the actual onset of symptoms. These types of reactions are, for the most part, initiated either by a CD8 cell or a CD4 cell. They have to be initiated by one of those 2 types of cells, and usually those cells are identifying the presence of some antigen that has stuck to the outside of a host cell, and so they begin to attack the host cell. I'll give you an example. If we look at haptens, remember haptens I told you were molecules too small to stick to an antibody in and of themselves, but once they stick to other larger molecules, then they can stick to an antibody. With poison ivy and poison oak, the poisonous chemical in that is a hapten. It's very tiny. Once it comes in contact with the surface of your skin, it can actually stick to things like collagen proteins in your dermis. And once it sticks to those collagen fibers, well, now it is identifiable by an antibody as a foreign agent. It's also going to be picked up by antigen-presenting cells, for example, and as a result, CD4 and CD8 cells are going to become activated. When they become activated, then those CD8 cells go out and start killing off any cells you have in your body that have poison oak molecules attached to them. Again, called delayed hypersensitivity because it doesn't happen immediately. It takes a few days for those haptens to stick to cells, for the cytotoxic T cells to recognize that there's a foreign agent present, etc. Let's talk for a minute about the concept of immunization. There are 2 types of immunization: active immunization and passive immunization. Immunization just means having some sort of molecules present in your body that have the ability to respond to some sort of infection. Sometimes those molecules that are present to respond to the infection were made by your own immune system, and sometimes they aren't. Sometimes we borrow antibodies, for example, from another person and use them to combat an infection that we're encountering. So the difference between these 2 terms are that in active immunization, your own immune system makes plasma cells, antibodies, B cell clones, T cell clones, cytotoxic T cells, memory B cells, memory T cells. Your own immune system is stimulated to respond, okay? Therefore, you end up having memory against that particular agent. In passive immunization, your own immune system is not activated. Not activated. Instead, we might have somebody inject antibodies from another person who's already been exposed to a particular illness, and they'll inject them into us, and those antibodies will help us deal with the infection. But because my own immune system wasn't activated, I have no memory cells which means if I ever get exposed to that same pathogen again, I'm still not going to be able to respond to it. Now, active and passive immunization can be of two varieties. It can either be natural or artificial. So you could have natural active immunization. You could have natural passive immunization. You can have artificial active immunization. You can have artificial passive immunization. Let me give you the difference here. I'll give you examples of both of these. Natural active immunization would be when you yourself are infected. Okay, you're out on the playground as a kid, some other kid coughs on you, you end up getting infected with a rhinovirus, and then you go through all the stages of having symptoms, and your T cells and B cells proliferate and the whole routine. That's active natural immunization. It happened naturally in your environment that you were exposed to the pathogen. Artificial active immunization occurs when you, like this boy here, get a vaccination. You are exposed to weakened versions of a virus, and this is done intentionally in an artificial environment, in a hospital, in a clinic, wherever it might be. The end result is the same, though. You're going to still be stimulating your own immune system to identify that pathogen that's been introduced, to attack the pathogen, and to manufacture memory cells. You can have passive natural immunization. When it's natural passive immunization, this is what, for example, is happening here with this woman who is pregnant and this woman who is breastfeeding. In both instances, the mother has a number of immunoglobulins floating around in the blood from all the previous times she's been exposed to different infectious agents. Those immunoglobulin Gs have the ability to pass through the placenta and into the fetus, and they have the ability to pass in the breast milk to the baby. So in both instances, we are infusing that fetus or that new baby with immunoglobulins from the mother, which means that whatever mom has been exposed to in her life in terms of infections and she has antibodies for, she's sharing that immunity with her baby. And it's done naturally through placental transfer or through breastfeeding. Notice, though, that this is a passive type of immunization because the baby's own immune system has not been stimulated. The baby has not made any memory cells. We're just taking advantage of antibodies from mom's breast milk and from the placenta being passed to us. The artificial passive version occurs if you get a shot of antibodies. They do this, for example, for certain types of diseases when people are going to foreign countries against, say, for example, hepatitis. They'll do it for kids that have low antibody titers themselves. They'll just take a blood donor and they'll concentrate all the blood plasma with the antibodies in it, and they'll inject that donor blood plasma into that child, and as a result, anything that the donor has encountered in terms of infectious agents and has made antibodies for, that kid now has passively acquired those antibodies. Again, because their own immune system has not been stimulated, they have not made their own memory cells, and so this is a passive immunization process, and as soon as those antibodies degrade, which happens in a couple weeks' time or so, then they're right back where they started, being susceptible to all kinds of pathogens. Now, I mentioned this previously a little bit, but I want to remind you again that organ transplants present a unique challenge to the immune system. If somebody's kidney fails, for example, and they receive a donor kidney from someone else, it's really important to find a donor kidney that has MHC1 on the kidney cells that are as similar as possible to the recipient's MHC1 protein. If you give a recipient a kidney from a donor with a very different MHC1 shape, the recipient's own immune system, the T cells, are going to go crazy. And even when the MHC1 is pretty similar to the recipient's MHC1s, T cells, CD8 cells, can still identify the difference, and they will attack. They will do that touch kill on the donor cells, and this is why organs are so often rejected. There is a future therapy that might work down the road that they're developing for this that involves something called FAST and fast cell receptors, but for the sake of time in this class, I'm going to skip covering that. I should say one thing, though, about organ transplants. A primary way that we prevent organ transplants is after a person receives a transplanted organ, they are often given immunosuppressive drugs. And these immunosuppressive drugs do as the name implies. They inhibit the activity of things like T cells in hopes that those T cells will not attack the donated kidney and destroy it. Sometimes people have to be on immunosuppressive drugs for the remainder of their life to prevent their own immune system from killing the donated organ. Another class of immune disorders are the autoimmune diseases. And this is, by definition, autoimmune diseases are diseases where the person's own immune system begins to attack their body and cause damage. This might be because they produce antibodies against their own cells or cell parts. Those are called autoantibodies. It might be because their cytotoxic T cells have been wrongly activated and begin attacking cells in their body. I'll give you a couple of examples of autoimmune diseases. And one thing you should know about all autoimmune diseases is, generally speaking, they tend to attack women more than they do men. And the age at which autoimmune diseases usually have onset is during childbearing years. So, 20s to 40s or so. During the years that people are producing children or potentially could produce children are the years they are most susceptible to the development of an autoimmune disease. Men can get autoimmune diseases, it's just that women are more likely. Some examples include lupus, or lupus erythematosus is another name for it. This is an example of a type 3 hypersensitivity reaction, and it involves the person's immune system attacking their DNA, types of cellular components, and causing lots of inflammation, particularly in connective tissues, although not exclusively in connective tissues. Another example is shown in this picture to the right, rheumatoid arthritis. This is an inflammation of the synovial joints. And it's also due to an autoimmune deficiency where the immune system begins to attack the synovial cells that line the synovial membranes. It causes severe inflammation. It can cause major deformities of bones, such as in places like the hand, as you see here. Other examples you might have heard of, things like rheumatic fever, which involves accidentally destroying things like the mitral valve at the heart while responding to a bacterial infection. Type 1 diabetes, which involved the destruction of the beta cells that make insulin. Multiple sclerosis involves the destruction of the myelin sheaths that surround cells. It surrounds neurons. And Graves' disease, which you already learned about, which is a disease where the body makes antibodies that mimic TSH. Okay, I'm hoping I can get to the next slide here. There we go. Last thing I want to talk about very briefly is immunodeficiency diseases. These are when the person's immune system becomes so weak or fails and they end up dying. There are two ways you can go about this. You can either be born with some sort of genetic deficit that makes it not possible for you to have functional B or T cells. That's called severe combined immunodeficiency syndrome, or SCIDS. Okay, that's when you have a genetic abnormality that leads to no immune system function. Or you can have what's called acquired immunodeficiency syndrome, which is the same as AIDS, and that's due to infection by HIV, the human immunodeficiency virus. So how does HIV cause AIDS? Well, HIV is what's called a retrovirus. That means that it contains RNA, and when that virus injects its RNA into a host cell, the RNA has to first be reverse transcribed into DNA to be injected into the nucleus so that we can then begin transcription of viral particles and take the cell over and make viruses. So the reason it's called a retrovirus is we go from RNA back to DNA in this case. The HIV virus, when it infects people, it preferentially infects CD4 cells, helper T cells. Also, it will infect some macrophages and neutrophils, but principally it's CD4 cells that get attacked by HIV virus. And the problem with this, as you remember, is if I destroy the helper T cells, I've destroyed my checkpoint for the immune system. And so anytime I get an infection, I'm going to have B cells and T cells saying, hey, where's the helper T cells? I know there's an infection here, but I can't get the green light to divide until a helper T cell shows up and confirms. If there are no helper T cells around to confirm and give the green light, then those B and T cells are in a holding pattern and your immune system cannot respond sufficiently to an infection. Usually the incubation period for HIV virus can be several months to years. Modern medicines have allowed us to control the severity of symptoms and the rate of progression of HIV. If it's allowed to progress to its final stages, that's when a person actually develops AIDS or acquired immunodeficiency syndrome. And that's really where they've lost the ability of their immune system to respond to infections. They get sick at the drop of a hat. They become ill very easily, and they oftentimes will die from opportunistic infections that in a healthy individual you would never die from those infections, but because their immune system's compromised, they can't fight anything off. Also commonly seen among patients with AIDS, they develop a very rare type of cancer called Kaposi sarcoma, which is a cancer of connective tissues that surrounds blood vessels. Very rare type of cancer, but it is associated with HIV infection.